another topic, that is lipid metabolism. And we can start with the de novo synthesis of fatty acid. De novo, the Latin word meaning from beginning. And we can start the de novo synthesis of fatty acid. And we should know these things, that is the site, that is cytosol. And again, it is present in many tissues like liver, kidney, lung, brain, mammary glands, adipose tissue, etc. So the site, that is intracellular site, it is extramitochondrial or cytosolic. Again, it is present in many tissues also. And again, we know the immediate precursor for this fatty acid biosynthesis is acetyl-CoA. Acetyl-CoA. And this acetyl-CoA, it is formed from, we know, from pyruvate. Pyruvate. Again, from certain amino acids. And again from the fatty acids. And again, pyruvate, we know, that is formed from aerobic glycolysis from the glucose. So carbohydrate, fatty acid and amino acids are the main source for this acetyl-CoA. And we know this acetyl-CoA is so formed, can enter into TCA cycle. It can enter into TCA cycle, or it is utilized for the biosynthesis of ketone bodies, or it is used in the ketogenesis. And again, it is used for the synthesis of sterols and our target, that is fatty acids. So that's the main thing, that is the immediate substrate for this fatty acid biosynthesis, that is acetyl-CoA. And again, we know the product formed, or we will study about the synthesis of palmitate or palmitic acid. So the starting material is acetyl-CoA and the end product is palmitic acid or palmitate. Fatty acid biosynthesis that will take place in the cytosol. And we know the acetyl-CoA formed are all in mitochondria. So what do you have to do? This acetyl-CoA that has to be transported to cytosol. So the acetyl-CoA that is formed in the mitochondria has to be moved out to cytosol. And there comes the importance of citrate transporter and all. So we can see how this acetyl-CoA formed in the mitochondria is transported into cytosol. So acetyl-CoA as such, that cannot be passed through this membrane. So it first converted to citrate. This acetyl-CoA that will combine with the oxaloacetate and form citrate. And the citrate, it is transported by the citrate transporter or tricarboxylic acid transporter that is present in the inner membrane of this mitochondria. And again, it is carried to the cytosol. And the citrate, so reaching the cytosol, it will be cleaved into oxaloacetate and acetyl-CoA by the enzyme citrate lyase. So the acetyl-CoA is so formed that can be utilized for the fatty acid biosynthesis in cytosol. And the citrate formed is again converted to oxaloacetate, and from there it is converted to malate, and through this malate shuttle, it will shuttle back to the mitochondria. So that's what happens. And again, in the de novo synthesis of fatty acids, we have to keep one thing in mind, that is, there are two enzymes that is very important for this pathway, that is acetyl-CoA carboxylase, and fatty acid synthase complex. Fatty acid synthase complex. This acetyl-CoA carboxylase is not the part of multi-enzyme complex, but it is the rate limiting enzyme here. Rate limiting enzyme in the fatty acid biosynthesis. And it catalyzes the first reaction or first step, that is the carboxylation of acetyl-CoA. So this is very important, that is the rate limiting enzyme in this fatty acid biosynthesis, that is acetyl-CoA carboxylase. Again comes another enzyme system, that is a multi-enzyme complex, that is fatty acid synthase complex, and actually it is a dimer comprising of 7 enzyme activities, And this fatty acid synthase complex that I have explained in another video, otherwise it will be too lengthy. So you just go and check that video also, that is about the fatty acid synthase complex, how it functions and all. And so basically we know that is fatty acid synthase enzyme have 7 enzyme activities, and this is organized as a dimer. So the first step in the fatty acid biosynthesis, that is the carboxylation of acetyl-CoA. So we know that acetyl-CoA on reaching the cytosol, that is catalyzed by the enzyme. This enzyme, it is very important, that is a rate limiting enzyme, that is acetyl-CoA carboxylase, and converting this acetyl-CoA to malonyl-CoA. And again we know this acetyl-CoA that requires certain coenzymes for factors and all, and we can tell that is ABC, that is required for A for ATP, B for biotin, and C for carbon dioxide. So just remember like that, ABC. So that ABC is required for the activity of this acetyl-CoA carboxylase, that is ATP, biotin. Biotin we know that is a member of the B complex, and again carbon dioxide. So for the carboxylation, definitely carbon dioxide is required. So these ABCs are required for the carboxylation of acetyl-CoA to malonyl-CoA, malonyl coenzyme, that is CH3 C double bond or SCO, that is converted to CO, CH2 C double bond or SCO. So that is the first step. Now coming to the second step, that is second step we can be categorized as second A and second B. That is, at first this malonyl-CoA so formed, that will go and attaches to the ACP of the multi-enzyme complex. ACP, that is acyl carrier protein, and I already told you that is fatty acid synthase complex, it is having 2 monomers, that is a dimer, and this malonyl ACP or that the malonyl-CoA that is converted to malonyl ACP, and that will bind to the ACP of the one of the monomer, acyl carrier protein of one of the monomer of this fatty acid synthase complex. And it is catalyzed by the enzyme that is malonyl transacylase. So malonyl coenzyme A that is binding to the ACP or joining to this ACP, acyl carrier protein of the monomer of this one of the monomer of the this fatty acid synthase complex, and is catalyzed by the enzyme malonyl transacylase that is present in that enzyme complex itself, producing malonyl SACP. And there comes the release of coenzyme A, acetyl-CoA. This coenzyme A is released here. Coming to the next step, that is second B, we can see the acetyl-CoA, it is joining to the another enzyme of another monomer, that is condensing enzyme or ketoacyl synthase enzyme of the other monomer of this fatty acid synthase complex. So malonyl-CoA that go and attaches to ACP of one of the monomer of the multi-enzyme complex, and this acetyl-CoA or acetyl enzyme, acetyl-CoA that will go and attaches to the condensing enzyme of the other monomer of this multi-enzyme complex, that is fatty acid synthase complex. So while attaching, we know definitely coenzyme A is released, and it is catalyzed by the enzyme acetyl transacylase that is also present in the multi-enzyme complex as the condensing unit. 3 units are there, and the condensing unit we can see the acetyl transacylase also. So we can say for the biosynthesis of this palmitic acid, that is the fatty acid we are going to synthesize. Palmitic acid, 2 carbon units are added. See, 2 carbon units are added on each cycle as malonyl-CoA. So these 2 carbon units are added as malonyl-CoA for this biosynthesis of palmitic acid. Now let us see the third step. So here we can see this acetyl enzyme, that is this acetyl enzyme and this malonyl ACP condense. That is acetyl enzyme and the malonyl ACP that will condense to produce acetoacetyl ACP or beta ketoacyl ACP. So definitely this condensing enzyme is released. Again one carbon dioxide is released, and it is catalyzed by the enzyme ketoacyl synthase or condensing enzyme. And that is the first enzyme we know that is present in that multi-enzyme complex system, that is a condensing enzyme or ketoacyl synthase enzyme. CH3CO, again we can see CH2CO, ACP, that is the COH is released as carbon dioxide. and we will get the acetoacetyl ACP or beta ketoacyl ACP. So now we can move to the fourth step, that is acetyl-CoA formed by the another enzyme ketoacyl reductase. So the reduction occurs and that requires NADPH. So NADPH plus H plus producing NADP plus. So we have seen so many things, that is this de novo synthesis of fatty acid requires ABC, that is ATP is required there, biotin is there, and carbon dioxide is required, and then another coenzyme, that is NADPH is also required. So what happens, this acetoacetyl ACP so formed is get reduced producing beta hydroxyacyl ACP. So ketoacyl ACP is reduced to hydroxyacyl ACP, so that there is the introduction of OH, the beta position, the beta carbon. So in the beta carbon we can see the formation of CHOH, and that is catalyzed by the ketoacyl reductase of the multi-enzyme complex or fatty acids in this complex. And this beta hydroxyacyl ACP, it is in the next step, that is step 5. So in step 5 we can see the conversion of this beta hydroxyacyl ACP that releases one water molecule and get converted to enoyl ACP. So enoyl ACP, that is instead of OH we can see water is released from here, H from the CH2 and OH from here, and CH, and we will get one double bond in order to balance this carbon. So that is the enoyl ACP. So it is catalyzed by the enzyme dehydratase, that is a removal of water molecule. So dehydration occurs, that is water is removed, and this hydroxyacyl ACP is converted to enoyl ACP. We can see the action of enoyl reductase on the enoyl ACP. So this enoyl ACP, it is acted upon by the enoyl reductase introducing H, so that this double bond is cleaved, becomes CH2. And again this enoyl reductase, that is the reduction is happening here, and that enzyme requires the coenzyme NADPH. So NADPH plus H plus producing NADP plus and forming the butyryl ACP. So this double bond is cleaved, H is introduced, CH2, CH3, CH2, CO, S, ACP, that is the butyryl ACP. And this butyryl group we know that is having 4 carbon. And so let us see how many more carbons are added to form this palmitic acid. We know almost 6 cycles is required for the formation of one complete palmitic acid or one fatty acid, that is having carbon 16. So 4 into, again 4, in order to become 16 we know the remaining it is 12. And again we know by the addition of 2 molecules, 6 into 2, 12. So 6 more cycles or 6 more times these steps have to repeat. That is from step 3 onwards. All these steps have to repeat for 6 times, and total 7 cycles will be there in order to form a complete palmitic acid. And the last step, we know there is another enzyme, that is thioesterase or deacylase, that is present in the fatty acids in this complex, that will release this palmitic acid by the addition of water. Water is required there for the release of palmitic acid, which is having carbon 16. So that's about the biosynthesis of fatty acids. So as a summary, the net reaction is like this, that is 1 acetyl-CoA, again 7 malonyl-CoA, in sense that is I already told you these 2 carbons are added as malonyl-CoAs. So that is 7 malonyl-CoA, that 2 acetyl-CoA that is converted to, that is 2 carbon acetyl-CoA that is converted to malonyl-CoA in the first step. So that is each 2 carbons are added here as malonyl-CoA. So 7 malonyl-CoA, 14 NADPH, that we know 2 NADPHs are required for each cycle, so or each pathway, so 14. So 14 NADPH plus 14H plus, giving 1 molecule of the fatty acid, that is palmitic acid, plus 7 carbon dioxide, 14 NADP again, 14 NADP plus 8 coenzyme A and 6 water, H2. So that is the net reaction for the production of 1 palmitic acid from the 2 carbon acetyl-CoA, and again adding each 2 carbons as malonyl-CoA in each step, and total 7 times this pathway has to repeat, producing 1 molecule of palmitic acid.